This presentation covers the regulation of glycolysis and gluconeogenesis. Your learning objectives for this lesson are to be able to describe the key steps and regulatory features of glycolysis and gluconeogenesis, explain the role of aldolase in both pathways, distinguish aldolase isoforms by tissue specificity and function, and understand that how aldolase B gene expression is regulated by transcription factors. Glycolysis is the metabolic process that breaks down glucose into pyruvate and produces ATP, the energy currency in the cell. It is the initial metabolic pathway for cellular respiration. Glycolysis occurs in the cytoplasm of both aerobic and anaerobic cells and does not require oxygen. The reactants of glycolysis are glucose, NAD+, ADP, and inorganic phosphate. The products of glycolysis are 2 molecules of pyruvate, 2 molecules of ATP, and 2 molecules of NADH. Glycolysis costs the cell 2 ATP molecules to complete the process. The purpose of the investment phase of glycolysis is to prepare glucose for energy extraction by phosphorylating and rearranging the molecule. The first step of the investment phase is to use the enzyme hexokinase to phosphorylate the glucose molecule. The reason for this is to trap the glucose in the cell. Phosphorylated sugars cannot cross the cell membrane. Glucose and ATP react to form glucose 6-phosphate and ADP. 1 molecule of ATP is consumed at this point. During step 2, the enzyme phosphoglucose isomerase converts the 6-membered ring of glucose into a 5-membered ring of fructose. This is done because the 5-membered ring of fructose splits symmetrically more easily later in the process. This step is reversible and is not highly regulated, and no ATP is used. During step 3, the enzyme PFK1 adds a second phosphate to the fructose. This process further destabilizes the fructose, making it easier to split the molecule later. This step requires the expenditure of 1 ATP molecule to complete. This stage of the investment phase is the rate-limiting and highly regulated step of glycolysis. This step is activated by AMP and fructose 2,6-biphosphate. ATP and citrate inhibit this step. During step 4, the 6-carbon sugar is broken into 2 3-carbon isomers using aldolase. This reaction is energetically unfavorable and proceeds only because the products are removed later in the process. This step does not require the use of ATP. During step 5, the enzyme triose phosphate isomerase converts DHAP into G3P. This happens to ensure both 3-carbon molecules can proceed through the second half of glycolysis. ATP is not used during this step. This step is extremely quick and efficient. The end result of the investment phase is the consumption of 2 ATP molecules and the conversion of 1 glucose molecule into 2 3-carbon molecules. The second stage of glycolysis is called the payoff phase. The first step of the payoff phase is the oxidation of glyceraldehyde 3-phosphate, and inorganic phosphate is added to G3P. This step is a redox reaction. The oxidation of G3P is paired with the reduction of NAD+ into NADH. The enzyme involved at this step is G3P dehydrogenase, which removes hydrogen molecules. Step 2 involves the transfer of a phosphate group, 1,3-biphosphoglycerate, to create ATP and 3-phosphoglycerate. This is called substrate level phosphorylation. The enzyme used during this step is phosphoglycerate kinase. Step 3 is the conversion of 3-phosphoglycerate to 2-phosphoglycerate. The phosphate located at carbon 3 is transferred to carbon 2. The enzyme involved in this step is phosphoglycerate mutase. Step 4 is the dehydration of 2-phosphoglycerate, or the removal of water to form phosphenolpyruvate. The enzyme used during this reaction is enolase. Step 5 involves the transfer of a phosphate group from phosphenolpyruvate to adenosine diphosphate, or ADP. This forms pyruvate and ATP. This step, once again, is a substrate level phosphorylation and is catalyzed by pyruvate kinase. This step is irreversible in glycolysis. Moving on, we'll talk about gluconeogenesis. Gluconeogenesis is the metabolic pathway that synthesizes glucose from non-carbohydrate carbon substances such as lactate, glycerol, and amino acids. It occurs in the liver and the kidneys, helping to maintain blood glucose levels. Gluconeogenesis is the reverse process of glycolysis, except that it bypasses the irreversible reactions of glycolysis by using alternative enzymes. The substrates are lactate, glycerol, glucogenic amino acids, and pyruvate. The purpose of gluconeogenesis is to maintain stable blood glucose levels and ensure a consistent energy supply to cells, especially during fasting and in between meals. Step 1 is to introduce a carboxyl group to make oxalacetate, or OAA. OAA is a TCA intermediate. This step occurs in the mitochondria and involves the enzyme pyruvate carboxylase. Pyruvate combines with carbon dioxide and ATP to form oxalacetate, ADP, and inorganic phosphate. This step is regulated by acetyl coenzyme A, and the cofactor biotin carries the carbon dioxide. Step 2 occurs in the cytosol in humans and uses the enzyme PEPCK. Oxalacetate is converted into malate or aspartate. This conversion allows the substance to exit the mitochondria. Once out of the mitochondria, the malate or aspartate is converted back into oxalacetate in the cytosol. This step is induced by glucagon and suppressed by insulin. Steps 3 through 7 are the reversible glycolysis steps, except they run in reverse. Steps 3 through 7 are generally grouped together due to this fact and other factors such as the absence of significant regulatory control steps and no net energy barriers. During step 3, the enzyme enolase catalyzes the reaction to turn PEP into 2-phosphoglycerate. In step 4, the enzyme phosphoglycerate mutase catalyzes the reaction that converts 2-phosphoglycerates into 3-phosphoglycerate. In step 5, the enzyme phosphoglycerate kinase catalyzes the reaction that uses 3-PG to create 1,3-biophosphoglycerate. This step uses ATP. In step 6, the enzyme G3P dehydrogenase catalyzes the reaction that creates glycerolhydride 3-phosphate from 1,3-PG. This step uses NADH. And in step 7, the enzyme triose phosphate isomerase catalyzes the reaction that creates DHAP from G3P. In step 8, aldolase catalyzes the reaction between DHAP and G3P to create fructose bisphosphate. Step 9 bypasses the glycolysis enzyme PFK1 and removes a phosphate group, moving toward the creation of a glucose molecule. The enzyme fructose 1,6-biphosphatase catalyzes a reaction in the cytosol that ATP and citrate activate. Step 10 is a completely reversible reaction that produces free glucose, which can be exported into the blood. This reaction occurs in the endoplasmic reticulum of the liver and kidney cells. The energy costs of gluconeogenesis are 4 ATP, 2 GTP, and 2 NADPH. Glycolysis is regulated through several mechanisms. Enzyme regulation is controlled by allosteric modulators such as hexokinase, phosphofructokinase, or PFK, and pyruvate kinase. Hormone levels in the bloodstream can also impact the metabolic rate of a cell. The concentration of glucose and other substances can affect the rate of glycolysis, and the levels of glucose can also enhance the efficiency of glycolysis. ATP levels influence the rate of glycolysis as well, ensuring the cell has sufficient energy to survive. Insulin promotes glycolysis and is released in response to high blood glucose levels. If we follow the diagram on the right, once the liver cells sense the high blood pressure, the high blood sugar levels, the pancreas releases insulin. The insulin will stimulate glycogen formation, which increases the glucose transporters and lowers the levels of glucose in the liver. When glucose blood levels are low, glucagon inhibits glycolysis and promotes glucose production. In this slide, we'll focus on the enzyme catalase, which plays a key role in step 4 of glycolysis. We'll cover the reaction it catalyzes, its reversibility, and its unique mechanism involving shift base formation at a lysine residue. Catalase catalyzes the cleavage of fructose 1,6-bisphosphate, or F1,6BP, into two 3-carbon sugars. They are glycerolaldehyde 3-phosphate, or G3P, and dihydroxyacetone phosphate, or DHAP. This occurs at step 4 of glycolysis. Only G3P continues directly into glycolysis. The DHAP, however, is quickly converted to G3P by triose phosphate isomerase in the next step. This catalase reaction is fully reversible under certain physiological conditions. In glycolysis, it breaks down that F1,6BP in gluconeogenesis. It catalyzes the reverse reaction, forming F1,6BP from the G3P and DHAP. The direction of the reaction depends on the substrate and the product concentrations. A shift base formation at a lysine active site occurs in type 1 atalases found in humans. The reaction involves the formation of a shift base intermediate. A lysine residue in the active site attacks the carboxyl group of F1,6BP, forming a shift base. This intermediate stabilizes the transition state and allows for the split of the C3 and C4 bond. The enzyme is then regenerated after the product release so that it can begin the process again. Human atalase is type 1, which uses a lysine-based shift-base mechanism. In contrast, type 2 atalases, found in some bacteria and fungi, use a metal ion like zinc instead of a shift base to stabilize the transition state. To summarize, atalase is critical in glycolysis for breaking down a 6-carbon sugar into 2 usable 3-carbon intermediates. Its shift-base mechanism is a classic example of enzyme catalase via covalent intermediate. The reversibility of the process makes it an important enzyme in both glycolysis and gluconeogenesis. Shift base. Atalase, especially the B isoform, plays a critical role in glycolysis and gluconeogenesis, so its regulation is tightly controlled by liver-enriched transportation factors. The atalase B gene contains several cis-regulatory elements in its promoter region. and serve as binding sites for transcription factors. These elements determine where, when, and how strongly the gene is expressed, especially in response to the metabolic needs of the cell. There are 4 transcription factors for Adelase B. HNF1 is a crucial activator that binds near the transcription start site. It's necessary for liver-specific expression of Adelase B. HNF3, also known as FOXA, acts as a pioneer factor. It can bind compacted chromatin and open it up for other factors to access the DNA. CEBP plays a role in mediating hormonal responses, such as insulin signaling, and contributes to metabolic regulation in hepatocytes. DBP, which stands for D-box binding protein, is involved in circadian rhythm regulation. It fine-tunes the expression of genes like Adelase based on the time of day, linking metabolism with the biological circadian clock. The transcription factors bind to their specific DNA motifs in the promoter region of the Adelase gene. HNF3 initiates chromatin remodeling, allowing HNF1, CEBP, and DBP to bind. This complex then recruits coactivators and RNA polymerases to form the transcription initiation complex. The gene's expression is modulated based on physiological signals like fasting, eating, and of course hormonal changes. Adelase transcription is a precisely controlled system that is controlled by a network of transcription factors responding to both internal and external cues, ensuring that energy metabolism in the liver is regulated at all times. The image to the right depicts the Adelase promoter region, the horizontal line, with labeled binding sites for HNF1, HNF3, CEBP, and DBP. Each of them has a different colored shape on the diagram so that they are easily seen. This diagram also shows their relative positions and interactions. You can see the HNF3 helping to open the chromatin, HNF1 and CEBP recruiting coactivators, and DBP linking circadian regulation to transportation initiation. In summary, we've seen that Adelase plays a dual role in both glycolysis and glucogenesis. There are key metabolic pathways that allow the body to balance energy use and glucose production. Among its isoforms, Adelase B is the liver-specific variant that participates in both pathways and is critical for proper liver function. We explored how its expression is tightly controlled by transcription factors like HNF1, HNF3, CEBP, and DBP. Each of those contributes to liver-specific and condition-specific gene expression. The regulatory networks ensure that Adelase B is expressed precisely when and where it's needed. Finally, understanding this regulation doesn't just help us grasp basic biochemistry. It's also crucial in understanding metabolic diseases like hereditary fructose intolerance, where Adelase B deficiency disrupts liver function. Altogether, Adelase serves as a model for studying enzyme regulation in a complex metabolic pathway.